Right.
Okay, so let me just quickly revise the structure of the chloroplast, because we will be needing these structures in the next slides. So as you know, there are two membranes, outer and inner. In between outer and inner membrane, there is intermembrane space. Within the inner membrane, whatever cavity is present, that is filled with stroma, and within the stroma there are stacks of a disc-like structure which we call as thylakoids individually, and the whole of the stack, like it is shown here, is called as granum, or plural is grana. And within this thylakoid structure there is a lumen, which is the internal portion of the thylakoid. So there is thylakoid membrane and thylakoid lumen, and lumen is the one which will have different proteins and so on. On the other hand, the stroma, which is outside the thylakoid, will have different enzymes and so on. So that way, this is the whole structure, and the stroma lamellae, which we are not going to study as such, but that is the stroma lamellae, which is kind of connection point between two thylakoid structure or two grana. So this is the whole structure of the chloroplast. Now what we have to look at is the actual photosynthesis process. Now so far whatever you would have studied would be like carbon dioxide plus water is equal to your glucose plus oxygen, right? That's the normal photosynthetic reaction that you would have written. But I had mentioned it earlier, the actual photosynthesis happens in two different phases or two different reactions, what we call as light photosynthetic reaction and dark photosynthetic reaction. So we will see one by one of these processes. So the thylakoid membrane actually houses the chlorophyll. So we know the thylakoid, which is this whole disc-like structure, the membrane of that is the thylakoid membrane. It is the one which will contain the chlorophyll pigment, and of course different protein complexes, including photosystem 1 and photosystem 2. Now, what is this photosystem 1 and photosystem 2? These are the complex of proteins, and it involves quite many proteins. We need not go into those details, which proteins, why are they required, what is their function, and so on. But we just need to know that these are the complex of proteins which help in the photosynthesis process. And they have some function related to the light absorption. So that is why the name itself, photosystem, uh, to them. So this thylakoid membrane contains chlorophyll and contains photosystem 1 and 2. And apart from that, it also has this enzyme which is called as adenosine triphosphate synthase. So any type of synthase is actually the enzyme which will synthesize that particular compound. So in this case, it is ATP synthase, means it is synthesizing the ATP molecule. You know, ATP is the energy of a particular cell, or energy house for a particular cell. It's not energy house, energy molecule of a particular cell, and that needs to be synthesized because there are so many processes that keeps on happening. So this ATP is synthesized in first in the cells, and that is present in the thylakoid membranes. And this we are specifically talking of the chloroplast structure. Obviously, ATP synthase is also present in all the different cell organelles or within the cells. But here we are specifically talking of the chloroplast. So within chloroplast, ATP synthase is present in the thylakoid membrane. And these photosystem 1 and 2, these are very specialized for the light dependent photosynthesis, which is first half of the photosynthesis reaction. That is what is called as light dependent, because it is utilizing the light and then doing the first reaction of photosynthesis. Now when the sunlight strikes the thylakoid, that is obviously sunlight will strike onto the leaf surface. Through upper epidermis, it will enter into the spongy mesophyll cells, from palisade mesophyll to spongy mesophyll, and within these cells, mesophyll cells, there will be chloroplast inside them. So obviously when the sunlight is passing through the leaf, it will reach this thylakoid membrane, and the light energy which is present in the form of sunlight will excite the chlorophyll pigment. Excite the chlorophyll pigment means, again, I would assume that you would have studied this in these parts of chemistry in those subjects. Like whenever you provide external energy to some molecule, it goes to an excited state. Excited state means it is higher energy state. So chlorophyll pigment, which is a protein or not protein, it's one of the biomolecule. I'm not sure if I have shown you the structure of chlorophyll pigment. So let me show you that part. Chlorophyll structure. Yes, I have shown most likely, right? So this is the kind of chlorophyll structure, which is a very complex molecule. And you remember last time we had seen it needs magnesium and manganese for its action, right? So that is where it is required for. So this whole molecule, it is obviously it's like when you make the solar light fall onto this molecule, which is present in the thylakoid membrane, it will get excited. That means some of the electrons will which are present in the bonds of this whole molecule, they will reach to a higher energy state. That is what is excitation of these molecules. Here it is. No, where is it? Here. Sorry. So, so the light energy will excite the chlorophyll pigment, causing them to give up electrons. So now, as soon as they reach to the higher energy state, electrons can escape from the orbit, right? That is the obvious phenomena that you might have learned in chemistry. So that way, when they reach to the higher stage, due to the light energy, they will get released from the chlorophyll pigment, and the electrons which are released, which they enter into electron transport chain. Now, electron transport chain is one of the mechanism through which the energy is produced. So again, electron transport chain. Maybe this one, I'm not too sure. Yeah, but something like this. Let me find a better version of this. Yeah. Okay, so this is I assume this is mitochondrial membrane, and sorry, mitochondrial. What did I say? Mitochondrial only. Yeah. So this is a mitochondrial membrane where there are different protein complexes. So once the electrons are received from the chlorophyll pigment, they will enter into this complex, then they will pass on to this, from this to this. So there are several steps which we call as cytochrome A, B, C, and so on. So these cytochrome molecules will accept the electrons and they will pass it on, and they ultimately help in the formation of ATP in general at the end. So I don't know where you can see it in a better manner. Yeah, probably here. Yeah, so you can see here that from ADP, ATP is synthesized with the help of ATP synthase molecule. So, and whatever you are seeing is the mitochondria, and these are the walls of the mitochondria. So that is whole of this chain of reaction is called as electron transport chain, in which literally the electrons are transporting from one molecule to the other molecule. This is not part of the syllabus, but you need to know what the electron transport chain is. So, yes, Bhavya.
So how do they exactly transport? I mean, they're getting transported in each and every different types of protein sites. So how do they get transported?
So, again, what is electron transport chain is just the transfer of energy as such from one molecule to the other molecule. So, you know, bond formation includes two electrons from the neighboring atom if it is a single bond, and so on. And once that bond is broken, these electrons, they are released free, right? And that is what can get transported. So this is ultimately in a very subtle way, if I can say that, it is a bond formation, bond breaking type of reactions. So in one case, once the bonds are formed, then they are broken, electrons are released. These electrons will go to the next molecule. Again, the bonds are formed there. Then again they are broken and they will release the electron, and then they will go to the next molecule. That way they transfer from one molecule to the other one.
Okay.
So these electrons, they enter the electron transport chain, which are released from the chlorophyll pigment, and a series of reaction that ultimately drives the phosphorylation of adenosine diphosphate to adenosine triphosphate, which is ATP. Yeah, so that conversion I showed you at the end, that is from ADP to the formation of ATP. That is what happens at the end of electron transport chain. So the electrons which are released by chlorophyll, they will be entering into electron transport chain. They will get transferred from one molecule to another molecule, and at the end, phosphorylation, that means addition of phosphate molecule. So this is adenosine diphosphate. So there are only two molecules of phosphate. It becomes adenosine triphosphate. There are three molecules of phosphate. So from adenosine diphosphate, one phosphate molecule is added to it, and it becomes ATP, adenosine triphosphate, which is nothing but the phosphorylation reaction, that is addition of phosphate. So this is the end goal of the electron transport chain, to generate energy within the mitochondrial structure. ATP is one type of energy molecule that you people know of. But apart from ATP, there are several other molecules. Like for example, there is NADPH, which is nicotinamide adenine dinucleotide phosphate. And this hydrogen is used here for the reduced form. That means it has extra hydrogen along with it. So this nicotinamide adenine dinucleotide phosphate, or in short NADPH, that is also another energy molecule. So like ATP, when it releases it breaks down the bond, it will release certain energy. NADPH, when it breaks down into smaller component, it will again release energy, and that's how the energy is utilized by most of the molecules within the cell. So the summary of this slide is chlorophyll. They will get excited due to the sunlight, because thylakoids, they have chlorophyll pigments. Thylakoids are receiving the sunlight. So the sunlight will fall onto this chlorophyll pigment. They will cause electrons to release or excite and then release. These electrons will enter into electron transport chain. From there, it will have a series of reactions, and the end goal is formation of ATP, that is adenosine triphosphate. And similarly, this NADPH can also be synthesized. Now, any question here? And I hope you people are following this. This is little tricky to understand and keep it in the head otherwise. Okay, so now again, the first point explains the same thing that we have learned earlier. One point that I have not mentioned is this is also called as light dependent reaction. Light photosynthetic reaction, it happens in presence of light, sunlight. So that is what is also called as light dependent reaction. So you will see several such names for this light photosynthetic reaction, or sometimes they are also referred to as light reaction. So that is the light dependent reaction. Now the grana, that is the whole stack of thylakoid. These are membrane bound stack structures present inside the thylakoid, functions by gathering the light. Now what we have seen is one thylakoid, within one thylakoid there can be many chlorophyll molecules, and this chlorophyll molecule will release electron. Now when we think of a grana, one stack of thylakoid, imagine the amount of electrons that whole of that grana will release, right? So, and within one chloroplast itself there can be even furthermore many number of grana, and that way they will release lot of electrons. So they function First by gathering the light, and this function is done by this photosystem 1 and 2. So photosystem, these are the complex of proteins. So again, let me try to show you those molecules. This is like very complex molecules, photosystems into structure of structure. Okay, now I don't want to scare you people by showing you this, but no, this is better. So you can see this whole thing is called as photosystem 2. This whole thing is called as photosystem 1. And you can see there are individually many different proteins, right? Whatever is shown here as a disk in green and yellow color and so on, these are individual proteins. So it is a complex of proteins. Similarly, photosystem 1 is also a complex of protein, which ultimately gathers all the light which has been received by the thylakoid structure, and then it will collect that light, it will transfer it to the chlorophyll pigment, and it will excite the electrons, and then it will cause the further reaction. So whole of this part, it says the same thing that we had discussed earlier, so I will not repeat it. What at the end we need to know is this final chemical equation, right? So there is water, that water is broken down. So this reaction involves light photosynthetic reaction, involves photolysis. Photolysis is what is shown in this diagram. So open image here. So if you look closely to this part, yeah. So if you look closely at this particular part, so light energy is getting absorbed. That energy is utilized for breakdown of water molecule. Water splitting complex will go and split the water into hydrogen and into oxygen, right? Hydrogen is utilized for the formation of NADPH. Now you know where that H comes from. That is from this water molecule, half part. And the oxygen which is released at the end of the photosynthesis is coming from here. And water we already have studied in the absorption by root, where the water comes into the leaf. So that same water is getting split into H and oxygen, that is hydrogen and oxygen, with the help of light energy. This particular part is called as photolysis. So that is the photolysis part. So, and the reaction goes like this. So 2H2O plus 2 molecules of NADP plus 3 molecules of ADP plus 3 phosphate. So these phosphate, they will be added to this ADP molecule. It will form ATP. So from diphosphate, it becomes triphosphate. The hydrogen from this water molecule will go and bind to NADP. It will form NADPH, and oxygen is released. And that way the reaction is also a balanced. If you see 2 NADPH, here also 2 NADP, these 2 hydrogen, they are released in here. So that is the light reaction in presence of the photosystem 1 and 2, or in presence of chlorophyll pigment. That's what releases the electron. This release of electron is ultimately utilized in the formation of ATP, where the phosphate is added to ADP, that is diphosphate to triphosphate, and NADPH is also formed. So that is the complete light photosynthetic reaction. Any question here so far? It's too cool.
Yeah, I have a question.
Yes.
What exactly is ADP and NADP, and then like?
Oh, sorry, I missed on that part. Then thank you for reminding. So everyone knows this very well in a way. ADP, or let me show you ATP structure, right? So you know, in case of DNA, RNA, we have this adenine, guanine, cytosine, and thymine, right? So this is the adenine part, right? These 2 nitrogen containing rings, that is adenine. This is your sugar molecule, which is even present in your this is ribose sugar because there is hydroxyl, so that is pentose sugar. So uh, nitrogenous base with sugar with phosphate. This is your nucleotide that is present in your DNA. After addition of these two phosphate molecule, that is what becomes your ATP. That is adenosine triphosphate. Adenosine is this whole molecule, right? Adenine, nitrogenous base along with the sugar, that is adenosine and triphosphate. So that is this ATP. Now similarly, there is this nicotinamide NADPH. This one I will show. So similarly, there is another nucleotide, adenine again, and then there is this particular structure which is one of the derivative of one of the what we say nitrogenous base from the DNA structure, right? So these are already there. Sugar, that is ribose sugar, is already there. Phosphate molecules, they are already there. They are just arranged in a particular manner. Now this is specifically called as dinucleotide because this is one nucleotide, and this above O, this is another nucleotide. So that is when nicotinamide, so nicotinamide, adenine, this is adenine, dinucleotide because this is one and this is one, that is dinucleotide, phosphate. They are bound by the phosphate. They are bound by the phosphoester linkage, but this is the phosphate. So that is nicotinamide, adenine, dinucleotide, phosphate. So again, these molecules are also present within the cell. Upon release of this phosphate molecule, or even the addition and removal of hydrogen molecules in NADP to NADPH, that's when the energy is released. So these NADPH also acts as a energy molecule within the body. Does that clarify, Janvi?
Yeah, thank you.
Okay. So now just to brief you whole of this process here again. So we know this chlorophyll, they are present in the thylakoid membrane. So this green thing which is written here as a membrane is nothing but the thylakoid membrane. This is the lumen or inside the thylakoid, that is the inside the disc, and then outside is the stroma part. So as soon as the energy falls onto this chlorophyll pigment, what they will do from the membrane, they will excite the electron. Those electrons, they will enter into the electron transport chain. They will cause the release of hydrogen ion because it's like splitting the water, right? H plus and oxygen is being released separately. Whatever energy is generated in that ETC will ultimately be utilized in the formation of ATP. So this is kind of summary of this light photosynthetic reaction. So, okay, we can see the dark photosynthetic reaction. So the next part of this photosynthesis is this dark photosynthetic reaction. So in this, what happens is, now we have the energy molecules, that is ATP and NADPH. These are utilized for glucose formation. Now you should remember that our end goal is to form glucose in the photosynthesis reaction. So that glucose has to be formed. How will it be formed? With the help of obviously ATP and NADPH. So they are being used in the light independent reaction. So this reaction, which is dark reaction, it is not dependent on light. So that is why light independent, and it is obviously called as dark reaction of photosynthesis. So in this, carbon dioxide and water, they are assimilated. Assimilated means they are forming a structure together, into organic compounds, and these organic compounds are nothing but at the end it will be glucose. So that is the kind of whole goal of the dark photosynthetic reaction. So using carbon dioxide and water, glucose will be formed with the help of ATP and NADPH. Now the light independent reaction, that is dark reactions of photosynthesis, they are carried out in the chloroplast stroma. So this reaction, which was light photosynthetic reaction, happened within the thylakoid membrane and inside. Now the dark photosynthetic reaction happens in the stroma, and the rest everything happens either in the membrane or inside the thylakoid structure. So dark photosynthetic reaction, like I said, it happens in the stroma of the chloroplast. So this stroma has a particular enzyme which is called as rubisco. Again, you will have to remember all of these names because they are very, very important from the exam point of view. So this stroma has, like I had mentioned earlier, while we are looking at the structure of the chloroplast itself, there are different proteins and enzymes, and one of them being rubisco. Now, what is this rubisco? Which is 1-ribulose 1,5-bisphosphate carboxylase or oxygenase. Again, let me show you that rubisco structure. So this is the kind of, this is the rubisco structure, which is kind of a complex of protein molecule. Here it is the perfect one, which is a 3-dimensional structure, which is a protein structure. Okay, but this protein structure is associated with the other molecules from the chloroplast itself. So this stroma, it contains this ribulose 1,5-bisphosphate. Bis means 2 phosphate, so ribulose sugar has 2 phosphate group, and it has this component of either carboxylase or oxygenase. So that is when the last CO. So it is rubisco. So that is the ribulose 1,5-bisphosphate carboxylase or oxygenase because it carries out the carboxylation reaction.
So do you have to remember like the proper name for it, like the biphosphate carboxylase oxygenase?
Yes.
We
Yes.
need to have everything on the slide.
Sorry.
We need to know all the stuff on this slide.
Not all this stuff on this slide. I will tell you what you need to remember. But this part, rubisco, yes, you need to know the whole name. This part is little additional in terms because you may not be able to understand what is the function of this rubisco, where is this Kelvin cycle coming from, what exactly happens. So that's why I have added certain other names, like you can see it here, phosphoenol pyruvate carboxylation and so on. But I will try to simplify it. But some of the names you need to know for sure, like including this rubisco. So this enzyme, it catalyzes the first step of carbon fixation. Now carbon fixation means there is atmospheric carbon within the surrounding the leaf, and this carbon dioxide is getting fixed into certain organic molecule. Fixed as in it is entering into the formation of certain organic molecule. That is what is carbon fixation. In this case specifically, this formation of molecule is nothing but your glucose formation. So that carbon dioxide is getting converted into glucose. That whole process is carbon fixation. So rubisco takes care of first step. So let me show you. Okay, this is not so good diagram to explain. Let me use the other one. What was that? Dark photosynthetic photo. No, let me put the Kelvin cycle. Okay. So you can see, I hope this part is visible to everyone. Let me open image in new tab. This So that way I can zoom in. Yeah, this is better. You can see this carbon dioxide is atmospheric carbon dioxide because it is free. This is the first step, that transfer formation of this molecule, that is 3-phosphoglycerate. Again, this you need not know, I'm just telling it to you. This is the first molecule formation. So you can see 1 2 3 4 5 6, these are the 6 carbon atoms present in this molecule. So these are 5 carbon atoms. Within this 5 carbon chain, 1 carbon is added, it becomes 6 carbon long chain. That is the first step of the Kelvin cycle, which is nothing but the part of the photosynthesis process, right? You don't have to remember all of this reaction. I will talk about these things later. As of now, what you need to remember, the rubisco, which is one of the enzyme, is involved in the carbon fixation at the first step of the Kelvin cycle, right? So that is what is being said here. Rubisco catalyzes the first step of carbon fixation in the Kelvin cycle. It is also called as Kelvin-Benson cycle and so on. And this Kelvin cycle is the primary pathway of carbon transport in plant. Carbon transport, it says, because glucose is transported across different parts of the plants. So ultimately, carbon along with that glucose is also transported. So among so-called C4 plants, now this is something that you need to remember because there are different types of plant. C3 plant, C4 plant. Now what is this C3 and C4 plant? That also I will show it to you. C3 and C4 cycles. Not good. Is this better? No. I want that specific reaction. Now, this looks very complicated. See, I can try and explain with this itself. Yeah. Open. So this is the C3 photosynthesis. So the plant in which C3 photosynthesis happens, that is called as C3 plants. The plants in which C4 photosynthesis happens, that's what is C4 plants. Now what is the C3 and C4? So when the photosynthesis occurs, we had seen in this case, it was forming this 3-molecule compound, right? From 3-molecule, it remains 3-molecule, it remains 3-molecule, it becomes 5-molecule compound. Somehow, that again we need not go into the technicalities of it, but the precursor in between is 3-molecule compound, 3-carbon compound. So in this C3 plant, this reaction of Kelvin cycle happens with the help of C3 carbon. That's why this photosynthesis is called as C3 photosynthesis, and that's why the plants are called as C3 plant. Now in C4 plants or C4 photosynthesis, what happens is this Kelvin cycle is associated with a compound which is oxaloacetate and malate, which are C4 in nature. That means they have 4 carbons. You can see here 1, 2, 3, 4. So these are 4 carbon precursor molecules which are involved in this photosynthetic reaction, and then they form the glucose molecule. And that is why this reaction or this type of plants, they are called as C4 plants. So in short, what you need to remember is C3 plants, they have a precursor, that is the in-between stage, which is of 3-carbon compound. C4 plants, they have an intermediate step before glucose formation, which is of 4-carbon compound. And these are oxaloacetate and malate. So that is what is C4 plant. So coming back to this part. So in case of the C4 plant, the initial carbon fixation step with rubisco and the Kelvin cycle, they are separated spatially. Separated spatially means they happen at different spaces, right? And the carbon fixation, it occurs by the phosphoenol pyruvate carboxylation. So this was one of the compound, phosphoenol pyruvate. upon addition of carboxyl group to it, which happens in the chloroplast, and this chloroplast is located in the mesophyll of the leaf. That is what happens while the malate, which is the 4-carbon product of that process, is transported to the chloroplast in the bundle sheet cell. So this becomes again complicated. So let me say it here itself. You can see here all of the reaction happens in mesophyll cell, right? So it is not specially the reactions are not happening at different spaces. While in case of C4 plant, this is the mesophyll cell and neighboring to it is a bundle sheath cell, which is a type of cell within the leaf structure. So this reaction, mesophyll cell, will happen like this, and the bundle sheath cell will have this Kelvin cycle. And that's why it says the two reaction, they happens at two different spaces. While in case of C3 plant, that is not the case. So that is what is being said even in this slide. So it is transported to chloroplast in bundle sheath cells, and that's where the Kelvin cycle is carried out. So after carbon fixation, where that 3-carbon product goes to, in case of C3 cycle, it remains with the mesophyll cell, but in case of C4 cycle, it goes to the bundle sheath cell. That's where the Kelvin cycle happens. That's where the glucose will be produced. So that is the kind of dark photosynthetic reaction. Now, does anyone have any question? And I will try to show you a video which will explain the same thing as a revision, but that will happen in the next class. But as of now, does anyone have any question? And like I said, probably that this can get a little overwhelming. There are so many names, so many complicated names, I would say, and everything has to be remembered apart from this last part. Yeah. So let me just highlight that, because you don't have to really remember this part, but yes, a part of it, yes, you need to remember. Also, let me make that as clear statement. This is not something that you should know. But this carbon fixation process, you have to know it. Yes, Aditya?
So for like, could you just do it such that you could just add a bit of a note on the stuff that we don't need to know in the slides?
Why? What will happen even if you read these 2 lines?
Uh,
Not harmful, I must say.
no, not harmful.
And like, it's not that complicated as well. I have tried to simplify as much as possible. And also you will learn this in the later stage. So let it be there. If at all we have to remove it completely, I will remove it from this slide. Yeah. Okay. So let me finish this part and then we will stop for today. So this is the end goal of your dark photosynthetic reaction. So it is also called as carbon fixing reaction because it will fix the atmospheric carbon into the sugar molecule, right? So whatever ATP and NADP were generated in the light photosynthetic reaction, they will be utilized in the dark photosynthetic reaction. And the plants will capture the carbon dioxide from the atmosphere through the stomata. Again, this process we had seen, and it will proceed to the Kelvin cycle, or it is also called as Kelvin photosynthesis cycle, Kelvin Benson cycle. There are different names, or they are also called as C3 or C4 cycles and so on. So in Kelvin cycle, these ATP and NADPH which are formed, they will convert the 6 molecules of carbon dioxide into 1 sugar molecule of glucose. And that is what is the reaction written here. 3 carbon dioxide, 6 NADPH, H2O and ATP. So ATP will be broken down to ADP, NADPH will be broken down to NADP, and this glyceraldehyde 3-phosphate is formed. Now this is the 3-carbon compound, which is one of the precursor in the glucose formation, right? And from this there are different molecules being synthesized. Ultimately it will form the glucose. So that is the whole of the reaction in this case. Does anyone have any question here? You might ask where is the sugar molecule, but like I said, this glyceraldehyde 3-phosphate will be utilized. So that is what is this? Where is G3P? Yeah, here it is. So it forms PGA, phosphoglyceric acid, to G3P, glyceraldehyde 3-phosphate. From this, it will form this G3P. The phosphate from third is changed to the phosphate on first. It will form this RUBP, which is ribulose sugar, and from there again one carbon molecule is added. So it goes on in cyclic manner. So one molecule will go in the formation of glucose, one molecule is recycled, again it enters, again one molecule goes for glucose formation. So this cycle goes. Any question? Anyone? I will remove this part because this is not that I would like it anymore to keep it here. Okay, does anyone have any question? Sorry for that. Not really. Done. Okay, so I guess let's stop here for today. Tomorrow we will quickly. Tomorrow we have class. Yeah. Tomorrow we have class. So we'll revise this quickly and then we will start with the next part. Whom?